you for the organizer and having me. It's a real pleasure and the first face-to-face meeting really for me since the pandemic, which is really interesting. Keith has kindly introduced the title of my talk, and I just wanted to thank the people who have contributed to the work that I'm going to present here, highlighted in bold. Right, in particular, external collaborators, Katja Konisheva, who's now in psychology at Birmingham, who's inspired a lot of the early stuff that we've done, but also Ed Alonso from City and Greg Slabo, who is now at QML, as well as people in the group, Chris Weaver, Nico Bale, and some former group members. Okay, so the work that we're doing is in many ways related to single molecule science. So this can be charge transport through single individual molecule junctions. It can be single molecule detection using nanopores and nanopipettes. There are some other things as well which I'll not talk about today, or if there's time, perhaps at the end. Now, a lot of these experiments have certain common features, right? And they are generally easily automated. So the measurement is automated. We collect large amounts of data, right? And typically the data sets that we measure reveal some sort of, or contain some sort of complex behavior. They show large variance, right? And I believe that you have to combine all these to really get to profound physical insight from the data. Now, I was going to illustrate that with two examples, and then based on those, show you sort of a hope interesting idea on how we deal with the problems that we engage or that we encounter. So the first example is around resistive pulse sensing and single molecule detection. So this is current time data. You know, what's a resistive pulse sensor? So that's basically a liquid-filled cell, which is separated into two half cells using a thin membrane. And then there's a small pore here that connects these two half cells. And by small, I really mean it can be as small as 2 nanometers, or it can be as big as 200. We've got significant control over the size of this pore. Now, because this pore is so small, it tends to be, from an electrical point of view, the major source of resistance. So if you have two electrodes in this system and you apply a voltage, a current will pass through the system that is largely determined by the size of the pore, of course also the conductivity of the electrolyte and the bias. Now, a piece of DNA is negatively charged. If you bias this electrode here positively, then DNA will eventually move across this membrane through the pore and end up on the right-hand side. And again, because the nanopore is the largest source of resistance in this system, any change in the conductance of this pore, for example, because DNA is moving through it, will change the overall current that we are observing. So typically what we measure in this sort of little cartoon representation are current versus time traces. And for every time a piece of DNA translocates through this pore, we see a dip, right? And this is what we call an event. In the simplest case, when the DNA moves through this pore in a linear fashion, then we get this sort of rectangular pulse. But there's a whole range of other things we can observe. For example, if the DNA is folded, we see this sort of initial excursion here, right? If the DNA has knots, right, then every knot, depending on the size of the pore, can be picked up, right? So think of the Christmas decoration as carefully as you are in packing this away. The next year you take out the decorations all tangled up. And very much in the same spirit, DNA is a long piece of string, right? And it's got knots in various places, and you can detect the presence and location of these knots using a nanopore sensor. Or from a more analytical point of view, we can also detect proteins adsorbed to the DNA, so we can make DNA structures that have capture probes associated with them, and those capture probes can then be made specific to particular protein targets, for example, for inflammation or detection or monitoring or sepsis or various other types of applications. Now, so what a typical readout would look like is this one here, okay? So a current time trace, and each individual of these blobs here, or these spikes, these events, correspond to translocation of an individual molecule. You can then start to analyze that, okay? So you've got a magnitude here, you've got a duration. This is fairly easy to define when you have a rectangular pulse. If you have other things like this sort of DNA carrier structure with a capture probe hanging off the side, then the situation becomes a bit more complex. So this is an example of 3 of these events here, but now you've got an individual probe hanging off the side, and these sub-events that occur within the event structure now contain the analytically important information. So we might want to ask or determine whether a target has bound to this particular capture probe, how many probes there are, what the ratio of bound versus unbound probes is to get an idea of concentrations and things like that. Yeah, and what you see here, by the way, is an AFM picture of one of those carrier structures, and this capture probe is hanging off the side around here. Now, in terms of some general thoughts about the data, well, the noise characteristics of the IT data is generally quite well known and characterized based on the electronic behavior of the recording equipment and things like that. Events are fairly sparse. They occur at random locations, but generally you'd have about less than 1% of data points associated with events compared to data points that are background. And there's some control over that, but there's not too much you can do about that generally. Typically, people would use conventional threshold searches to identify events, okay, but it's not the only thing you can do. We're currently working on either statistical means of identifying events, for example, through zero crossing theory, or you could even think about a neural network that's trained up on noise, but then discovers that, for example, when it encounters an event, it's not very good at predicting that, so you could also use that, and importantly, potentially use that without making any great assumptions about what event shape you are expecting. Yeah? In terms of analytical applications, we typically know the types of events we are expecting, so the event classes are known. So, for example, this example on the left you've seen before, this is a carrier DNA with 1 capture probe and the target bound. And again, you notice the sub-event here. On the right, there's a carrier with 2 sub-events here and there, right, and so forth. So in principle, you can do control experiments that provide you with the training data. So that would be for supervised classification, but there is scope for unsupervised classification as well. So if you, for example, zoom into the entrance of the pore, then initially the DNA is in solution, it's subject to Brownian motion. It then comes close to the entrance of the pore where there's a strong electric field, and then it's forced to unravel as it goes through this little pore. Now, what we tend to see in a translocation event is this transfer in this in the narrow part of the pore. But actually what happens on the outside as the DNA approaches the pore and undergoes certain dynamics before entering the pore is actually something that no one has really looked at, and I don't think we actually have the tools to study that. Right? This would all happen at the onset of this event here, right? And you might be interested in changing noise behavior or some other patterns. So that's one example. The second example is single molecule conductance data. Now that's not so much for sensing, but more for molecule characterization. We use scanning tunneling microscopy for that. And if you've heard of STM, you will know it as an imaging technique. And what you see here is, on the left is an image of gold atoms in an acetate buffer, or on the right-hand side, this is an organic add layer on gold. And you get very high atomic or submolecular resolution using this technique. But you can also use it to identify, to probe individual molecules. So, for example, you've got this 4,4' bipyridine on the surface. You approach that with the tip of an STM, which is basically a sharp needle. The tip initially is pushed into the surface and then withdrawn. And as it withdraws, it actually picks up this molecular species on the surface until the junction actually breaks. And when you look at the current as a function of as a function of distance, then you can normally, you'd expect a behavior like this. So initially a decrease, then there is some plateau feature while the molecular junction is intact, and then it breaks and drops to what was often noise level. So you can extract the current per molecule from the step height here. You can also identify the length of a molecular junction before it breaks. So this is all quite useful. Typically, you would record, let's say, thousands of these data traces, and then you produce either a 1- or 2-dimensional histogram. And the result of well, one example is shown here, and you can see here this, for example, is the plateau feature, you know, and initially this is full contact, and then the junction breaks off as the gap becomes larger. Now, what you can also see here, and I'll just point this out, this is the conductance on a log scale, right? And this is the distance, and you see how much variation there is in the data. Even if the 2D histogram suggests there is a certain feature here, there's quite a lot of variance, and this is, across this plateau, this is about a factor of 10 in conductance. Yes, and that's reminding me of a talk we heard yesterday, right, and how the changes in the local environment actually change the overall performance of a junction. If you then investigate this in a bit more detail, you actually find that the plateau feature that you would expect isn't necessarily what you observe, or at least looking at the entire data set, there's a lot of other behavior. And I should point out this is a different data set, this is our own, but what you can see here is, well, something that's similar to a plateau, but more of an undulation. Then you've got one that seemed to undulate, then break off. You've got telegraphic noise patterns and so on. So a variety of behavior there. And you know, some of this might be artifact, but some of this will have physical meaning. And what we really want to do is extract all that information and be able to interpret that. So especially for new molecular systems, it is, first of all, not entirely clear what sort of feature we would expect in the data. And it's not even advisable, perhaps, to make any assumption there, because the expected behavior, well, that might, for example, create a user or confirmation bias, because in a large variable data set, you might just find what you're looking for. Okay? It could also lead to information loss, because you might not find what you aren't looking for, in a sense. Yeah? And you miss the complexity of the data set. Okay? So you might miss the presence of subpopulations, and you might be unable to quantify them if you just use, say, 2D histogram conventional approaches. Now, a dimensionality reduction is, of course, something that can help here, and we've played with a range of different methods in that context. And I'll not talk about all of them, just pick one example, and that is the unsupervised data classification using image recognition combined with dimensionality reduction. And this goes back to an idea a former postdoc of mine had, Anton Vladica, who had done experimental work with Michel Calamy in Switzerland, which they had published. And when he joined my group, we then had a look at the data and see whether we could learn something from further analysis. So this is then published in here, Machine Learning in Science and Technology. And an example of a single conductance-distance trace is seen here. You can see the sort of recognizable plateau features and so on. And his idea was basically, well, if we look at this data, can we look at this data as an image and use effectively, well, neural networks that have been trained on image recognition to extract physically meaningful features from the data. So we then set out and used things like AlexNet, ResNet, or VGGNet. As many of you will know, they have already been trained on millions of photos of unrelated objects, so dogs, cats, ships, and so on. And, well, what they then, once trained, they can, of course, identify unseen images and what's on them. And again, what we'll then wanted to see is whether we can use the training for this non-domain specific data to identify things that are physically meaningful in the data that we have. Right, so what we've done basically is to use AlexNet for most of our work. And the way this tends to work is, so this is the overall structure. AlexNet has got a sort of a feature extractor part, and it's got a classifier part. And just in broad strokes, you put an image in this case of a dog, right? It produced a reduced dimensional representation of this image, and then classifies what it thinks is on the photo. Now, we've changed the network. We've basically replaced the classifier with a dimensionality reduction part. In this case, I'm showing an autoencoder, but we've played with other things as well, which then takes the feature extractor output and reduces it further to something like a two-dimensional representation where different molecular behavior would then be represented by different clusters. Again, I'm just going to give you an example here. This is a recorded data from previously published data, 3 molecules. It's essentially a molecular wire which is connected to electrodes here and here. we call them BDNC, MBDNC, and TBU-BDNC. That kind of means something to the chemist, really, because that it's just about the change of these R groups here. But if you look at the original output, you'd see that in terms of 2D histograms, BDNC and MBDNC, for example, show overall different behavior. You get different plateaus, different numbers of plateaus, and so on. Now, when we then use the raw data in conjunction with dimensionality reduction techniques, and if you just look at PCA, for example, here, you'll see that all three molecules are kind of separated, but not really, right? They fall into different areas of the cluster plot, but we don't see distinct clusters with them, and that was the case also for TSNE, or the autoencoder applied to the raw data. Now, when you look at the individual molecules, and I should say they were measured separately, but they were, for the purpose of this analysis, the data was pooled. Yeah? If you look at the results, overall, in none of the cases that we look at here, we see different behavior or subclustering, right? There's no subgroup within each molecule, unless you, ignore the black dots for now. Come back to that in a second. So based on that, you would expect that each individual molecule is represented by a single ensemble, right? Now, then we thought, okay, well, let's see what the feature extractor can do for us. So we now use an autoencoder on the feature extractor output of AlexNet. Okay. Again, an example of the trace for MBDNC. And initially, the result was somewhat disappointing. So we've had 3 different molecules. Okay, they all kind of, overlap to some extent. But what's then interesting is that if you look at the MBDNC data set, after feature extraction and dimensionality reduction, we all of a sudden find 2 clusters, so 2 subpopulations in the data. Yeah, we've got the red majority one, which turns out when you look at the 2D histogram, you see this sort of behavior here. And then we've got the brown one, cluster 1, which shows behavior that's somewhat different. It actually shows a slightly lower plateau, and it shows a much shorter drop-off. Yeah, and when you analyze the plateau position, for example, in a bit more detail, you find that the conductance ratio is about 2. Yeah, and that the nature of this drop-off being very sharp in cluster 1 led us to the hypothesis, at least, that perhaps in cluster 1 you've got individual single molecule junctions that break off cleanly, right? Whereas in cluster 0, which in this case was the majority cluster, you get slightly higher conductance, so maybe 2 or more molecules, which then don't break off very nicely, very clearly. Yeah? We then looked at the effect of training in this case. So for all 3 molecules, you see here, this is for 100 epochs, 300 and 1,000 epochs training of the autoencoder. For BDNC, it actually doesn't make a difference. We've checked until 10,000 epochs. There was no new cluster formation. MBDNC, this is the one that you'd seen before, right? After increased training, you get better separation. And then interestingly, for the TBU-BDNC, after increased of training, we then also started to see a subpopulation here, okay? Now, I don't want to go into great detail and what the implications are there, but just draw some conclusions to this point. So we've reanalyzed charge transport data for 3 molecules that have previously been characterized. The dimensionality reduction on raw data as well as on feature extractor output from AlexNet has been performed, and in the latter case, well, we don't see any subpopulations for BDNC, even for extended training. We do see subpopulations for MBDNC and TBU-BDNC, right? And this was previously unknown and unexpected. There was no obvious reason why they would form subpopulations. Now, the physical interpretation is, of course, difficult and I would say tentative so far, right? But we could, there was a sort of new insight, if you like, into the behavior of this system. And perhaps a more general note is that the feature extractor in this process, it was not retrained, right? It was just used as is. There's no domain-specific knowledge in the feature extractor, and in this case, it actually didn't have to have any. Right? We'd like to think that it still picks up physically meaningful differences between the data. And then maybe final couple of minutes, I just wanted to show you some new results where we use a similar concept, now not applied to single molecule charge transport data, but voltammetric data, electrochemical data. And if you're not an electrochemist, I'll just explain briefly to you what this is. So voltammetric data are data where you measure a current as a function of applied voltage or potential. The shape of these data traces then depend on the reaction mechanism of the reaction that you're investigating. It depends on the electrode geometry and the size. So when you look on the left-hand side here, there are three different voltammograms shown at the top here. One is for a reaction that follows a simple electron transfer mechanism. The second one in light blue is an electron transfer reaction plus a chemical reaction. And then in dark blue, it's an electron transfer, chemical reaction, electron transfer, right? So a coupled system. And you see how the shapes differ, right? And how they also change as you change the electrode size. If you ask where this could be applied, well, potentially, for example, in electrochemical surface imaging, where you map out reactivity in different places along the surface, or perhaps in quality control and testing of autonomous electrochemical sensors. Yeah? Now, the workflow is similar as before. So we've got the electrochemical data. We plug them into this workflow. We either use dimensionality reduction on the raw data set, so this is just the image data, or we put it through a feature extractor, and in this case, we use BGG16, right? And then we sort of perform dimensionality reduction on the features, right? And this is a toy example here, but this triangle, for example, would correspond to the three mechanisms for a given electrode radius. Okay? So in terms of a classification task, so for every electrode radius, we get three CVs, E, EC, and ECE, and we've got 6 different radii. We try to perform the classification using dimensionality reduction techniques, and we've used PCA, TSNE, and UMAP, okay, either on raw data or feature extractor output, okay, and the objective here is to both, in a sense, maximize the separation between points within a triplet. We use a distance metric here, or between groups, right, between different electrode radii. In this case, we use a silhouette score. Right, and as I said, in the interest of time, I'm just going to skip ahead. Now, what are the key outcomes in terms of these two metrics? Well, high values is good separation, right? And whatever is blue here refers to raw data, and whatever is orange refers to features. Okay, and if you look at the left one here, so this is the separation within triplets. Okay, and you basically see that it doesn't really make much of a difference whether you apply dimensionality reduction on features or on raw data. But actually that turns out to be very different when you then look at the separation of triplets, so according to electrode radii. So it's quite a differentiated picture, right, where PCA does a lot better than on features compared to raw, right? And it also does better compared to TSNE, for example, which in the case of the within triplet separation, TSNE was better. So it's really a question of looking very carefully at which method we're using for what purpose. For the PCA analysis, we also find that we looked at the variance explained, and we also find there that PCA on features, well, basically leads to compression, if you like, of variance to the first 3 dimensions. So the first 50% of the variance is, within the first 3 dimensions in this case, whereas for PCA applied to raw data, it takes about 6 dimensions to capture the same variance. So to come to the final conclusion, we've classified voltammetric data using dimensionality reduction on raw data and feature extractor output. To differentiate different mechanisms, so within triplet, right, we see little difference between on raw data and on features, right, but we do find that TSNE is better than UMAP and better than PCA in that case. To differentiate electrode radii, so between triplets, PCA, TSNE, and UMAP perform better on features across the board. For PCA, we find more variance condensed to lower dimensions, so you could say the information content is more compressed. And on features, PCA is better than TSNE, which is better than UMAP. Okay? And as before, there's no domain-specific knowledge, and separation is based on shape, on appearance. And I would argue that maybe there is some generality to this approach. Now I will stop here, and thank you for your attention. Happy to take any questions.